Data visualization of a full-term placenta dataset using R. A little bit of background on our data is that the metadata came from Olney et al. At the Wilson Sex Chromosome Lab, and it was used to identify differentially expressed genes between the sexes. We are taking from the batch 1 data, and so there were 22 total observations, 10 of which were female with XX sex chromosomes, and 12 of which were male with XY sex chromosomes. And RNA-seq analysis was performed on the full-term placenta, and the tissue samples were taken from white, black, Asian, and other reported races. One thing I noticed in the manuscript was that there was a claim made that males born from mothers with obesity were more likely to be overweight at 1 year of age than females born to mothers with obesity. So what that led me to question with the data set that we had, right? We are using placental tissue. We do not have any samples from 1 year of age. So the way I thought we could contextualize this with our given data set was to ask if the maternal pre-pregnancy BMI influences the weight of the males and females. So looking into the research question, the way I set this up was to be able to ask 3 smaller questions that helped me assess if my data set was enough to even tackle this question, and B, actually test to see if I noticed any correlation between the pre-pregnancy BMIs and the birth weight of the males and females. So the 3 questions that I used are, what are the distributions of pre-pregnancy BMI considering sex as a variable? And the method I used was the histogram. The next question I used was the distribution of birth weight considering sex as a variable, and for that I used a violin plot. And for question 3, is maternal pre-pregnancy BMI significantly associated with birth weight for male and female samples? And for that method, I use scatter plots. So if we look at the data, so I went ahead and I summarized it using the SCIM-R package, and you can see that we have different types of variables. So the first ones being the samples, we have 22 unique samples, we have 2 different sexes, and we have 4 different races as we outlined before. And then also other variables to look at, these numeric variables. So we have, the maternal age, and, we had an average of 33.3. Gestational age, that's how many weeks. You have gravidity and parities. Pre-pregnancy BMI, that's the one we're concerned with. So pre-pregnancy BMI is the one where we can see there is some variation if we look at the histogram plot. And we have a mean of 26.8, so that's already higher than 25. So, the average mother that was in our sample was overweight. And then for birth weight, the average birth weight, again, we have a good distribution, was about 3,405 grams. So looking at the distribution of the pre-pregnancy BMI, considering sex as a variable, so making sure that I do have male samples and female samples within the different BMI ranges, right? We want to have those normal ranges, but we are looking for evidence of these higher ranges too. So this helps me get a breakdown. I was able to put our line here to show us the threshold for where overweight starts. And from there we can see we do have a fairly good distribution of BMIs across, normal to obese. Next, I wanted to investigate the distribution of birth weight considering sex as a variable. The median birth weight was about 3,300 grams for females and about 3,450 grams for males. And the range of female birth weights, as you can see, was much narrower than the wide range of males. So the female range was about 2,500 to 380 grams, and then the male range was about 2,600 to about 4,600 grams. What we did was we created scatter plots with the independent variable of maternal pre-pregnancy BMI versus the dependent variable of birth weight in grams. And we used facet grid to split those up via female and male for readability. And looking at this right away, we can see that there is definitely a positive correlation on the side of the female and what looks to be a negative, almost negative correlation on the side of the male. So just by looking at this, it's almost the opposite of what we predicted. We predicted that there would be a correlation with the males in the study, and instead we're seeing the reverse. And now to test that, we ran a multilinear regression model. And so looking at it, if we're looking at the birth weight as a function of pre-pregnancy BMI and sex, we see that the intercept is the expected value of the birth weight when we're at our reference level, or in this case, since we have sex male here, we know our reference level is female. And it appears, like we had noted, that it is significant. We lose that significance when we're looking at pre-pregnancy BMI and sex male versus the birth weight in grams. So what does that mean, we conclude? So in terms of our question regarding maternal pre-pregnancy BMI influencing the birth weight of males and females, there was enough evidence to suggest that maternal pre-pregnancy BMI correlated with female birth weight, but there was not enough to suggest that it correlated with male birth weight. This could be because of our sample size. This could be because of our sample distribution. We could add more individuals from another batch later on to see if that changes our results. But one thing I thought would be interesting would be to evaluate the BMI of patients at 1 year of age and to be able to reevaluate the claim and compare them and their progress from the full-term placenta.